Hello everyone again. So we will continue apoptosis. So in the previous video, we studied about what is apoptosis, uh, what are the major factors involved in apoptosis pathway, then general signaling of apoptosis. So in this video, we will again continue apoptosis pathways. So basically, uh, we have already discussed the general signaling. So we will continue with the pathways. So there are actually two pathways of apoptosis, intrinsic pathway and extrinsic pathway. Clear. So the one that you are seeing on the slide is the intrinsic pathway. So intrinsic pathway basically means that the death stimuli or the signal, which the cell death signal actually is generated inside within the cell only, and uh, after the signal is generated, apoptosis pathway is initiated by all the three major factors that we covered in the uh, last session. That is caspase, mitochondria, and our BCL2 family proteins. So extrinsic pathway is somewhat different from intrinsic pathway. So in extrinsic pathway, so signal is not generated within the cell. It comes as a ligand or a signal comes from different place or is produced by different types of cell, and the signal will come and bind to the receptor on the plasma membrane, and after that the signal actually uh, activates our extrinsic pathway. So we will see both the pathways in detail. So first we will start with intrinsic pathway. So just like I mentioned, intrinsic pathway means, suppose this is a cell. You are saying this is a cell. This is the plasma membrane you can see, and this is the cytosol portion, and this is our nucleus. Okay, nucleus, and inside the nucleus we are having our genetic material, that is a DNA, and you can also see the mitochondria. Since mitochondria is the major factor involved in apoptosis pathway, so in intrinsic pathway you see the role of mitochondria. So that's why sometimes intrinsic pathway is also known as mitochondrial mediated apoptosis pathway. Okay, so yeah, this is a picture of a cell, and yeah, nucleus, mitochondria, and yeah, just like that you can see some receptors also present. Okay, now intrinsic pathway, I already mentioned the pathway. This pathway, in this pathway, the stimuli or death stimuli or apoptosis stimuli is generated within the cell. That means the death signal will be produced inside the cell only, and after that signal is initiated, yeah, mitochondria mediated apoptosis pathway will be activated. So let's see here intrinsic pathway where the stimuli is formed internally in the cell. So you see one, two, three. So there are three cases mentioned in this uh, picture. So let's talk about the first picture. Okay. So how intrinsic pathway is activated or how the stimuli is generated in the cell itself. So the first case is the signal will be generated within the cell if there is absence of growth factor or trophic factor. Here you can see it's actually a growth receptor or growth factor receptor, which actually binds with the growth factor. Okay. Suppose if a growth factor is present in the cell, okay, if this one you can see trophic factor receptor and trophic factor or the growth factor, when it binds to its receptor, when it's bind to its receptor, the receptor actually dimerize, and yeah, that receptor will activate further pathway. And this pathway, pi3 kinase pathway, is called our our protein kinase B signaling. And whenever the growth factor is present, yeah, this our PKB, protein kinase B signaling, will be activated, and this protein kinase B always, you can see it phosphorylates our BH3 family protein called BAD, and phosphorylated BAD is always captured by a 14-3-3 adapter protein. Okay. So in the presence of growth factor, cell will never try to kill itself by intrinsic mechanism. That means the growth factor will always come to its receptor and will keep the signaling on, protein kinase B signaling on, and the protein kinase B always phosphorylate our BH3 only protein bad. And we know that BH3 only family protein always bind with anti-apoptotic protein, thereby freeing pro-apoptotic protein to activate our apoptosis pathway. So whenever the cell is growing, growth factor will be released, and growth factor will always keep binding on its receptor. And with the help of that signaling, yeah, BH3 family protein bad will be always phosphorylated, and phosphorylated bad will always bind with a 14-3-3 adapter protein. Thus, our BH3 family protein is not able to bind with anti-apoptotic protein. So, anti-apoptotic protein will always bind with our pro-apoptotic protein, not letting them oligomerize. If they don't oligomerize, yeah, our cytochrome C will not escape from mitochondrial intermembrane space, and it will not be able to bind with APAF1, and yeah, the caspases will not be activated. So, when the growth factors are present, apoptosis never takes place. Now, this is the first case. Suppose if the growth factor is absent or the growth factor is not coming. When the growth factor is absent, at that time, cell tries to kill or activate intrinsic pathway. So, this is considered as a stimuli, death stimuli. When a growth factor is not coming to a cell, that cell should be killed. And that's what happens whenever the growth factor will be absent. The growth factors will not come and bind to its growth factor receptor. Thereby, this protein kinase B signaling will be inhibited. So, protein kinase B will not be able to phosphorylate our BH3 family protein called BAD. And you know, BAD is a BH3 family protein. It will go and it will bind with anti-apoptotic protein, either BCL2 or BCLXL. Thus, anti-apoptotic protein and BH3 only family protein bind to each other. Now, by that, our pro-apoptotic protein monomers, they oligomerize and they form a channel, and through this channel escapes our cytochrome C. And we know cytochrome C will go and bind with APAF1 adapter protein, forms a death wheel. It will activate initiator caspase, and initiator caspase will cleave the pro domain of effector caspase, and effector caspase will cleave the cell substrate, finally leading to the cell death. So this is the first case. Here, what was the death stimuli? The death stimuli was absence of growth factor. So when a growth factor is absent, that is actually a sign or a signal to kill that cell. Similar way, second type of death stimulus is produced in the cell when the DNA of that cell is heavily damaged, maybe due to some carcinogens, or maybe when DNA is exposed to harmful UV rays, DNA becomes mutated or DNA becomes damaged. At that point, we know that there is a tumor suppressor angel gene called P53. So whenever DNA is damaged, P53 gene becomes activated and transcribes it transcribes, or it transcribes puma. Okay, it transcribes the puma protein. And just like bad, puma is also a BH3 only family protein. So yeah, noxa is also produced when DNA is damaged. So when DNA is damaged, at that time, puma and noxa are produced, and they come out of nucleus and they go and do the same thing. This puma and noxa are BH3 family protein, so and they are only activated during DNA damage. So puma and noxa will come out of nucleus, and they will go and inhibit anti-apoptotic protein again. When anti-apoptotic protein is bound to Puma and Noxa BH3 proteins, pro-apoptotic protein again becomes free. They oligomerize and they form a channel-like opening. Again, cytochrome C can come out from mitochondrial intermembrane space and they will bind with adapter protein called APAF1 forming apoptosome death wheel, and as usual, APAF1 will activate initiator caspase, and initiator caspase will activate effector caspase, and effector caspase will cleave all the cell substrate and the cell will die. So, in the second case, the death stimuli was DNA damage. Okay, if it is a small damage or small type of damage which can be easily repaired, our DNA repair mechanism can easily correct it. But until the DNA is heavily damaged and it is beyond repairable, yeah, cell will try to activate apoptosis pathway and kill it. Okay, so during heavily DNA damage only, Puma and Noxa will be produced or transcribed by P53 gene, and they will, as usual, go and inhibit the anti-apoptotic protein, and pro-apoptotic protein will oligomerize, form a channel, let the cytochrome C escape from the mitochondrial intermembrane space to cytosol, and once cytochrome C binds to APAF1, it will activate initiator caspase, and initiator caspase activates effector caspase, and that leads to cell death. Okay. Now, another another method, that is the third scenario where integrin molecule is disrupted. So integrin is actually a cell to cell adhesion molecule and it helps in cell migration and cell motility. And if integrin is disrupted, cell cell to cell adhesive molecule integrin is disrupted due to any case, at that time a BH3 family protein called BIM will be activated in that case. So BIM is normally found on the microtubule cytoskeleton. So whenever integrin is disrupted, BIM detaches from our microtubule, and yeah, it's a BH3 only protein. So again, it will do the same thing. It will go and it will inhibit anti-apoptotic protein by binding it. As usual, again, anti-apoptotic protein will not be able to bind with pro-apoptotic protein. So bags or back pro-apoptotic protein, they oligomerize and they form a channel-like opening. And from this channel-like opening, you can see cytochrome C will again escape from intermembrane space of mitochondria to cytosol, and cytochrome C will be binding with our adapter protein APAF1, and APAF1 will again activate initiator caspase, and initiator caspase will cleave the prodomain of effector caspase, and once the effector caspase is activated, it will go and it will cleave all the cell substrate, leading to, yeah, cell death. So here, this is the third scenario, which is disruption of integrin signaling. So that is about our intrinsic pathway of apoptosis, where the death stimuli is generated within that cell only. So we saw the three scenario where growth factor was missing. That is also an apoptotic stimuli. If integrin is disrupted, that is also a death signaling or apoptotic stimuli. Then the third one that we saw is heavily damaged DNA. When DNA is heavily damaged, that is also considered as apoptotic stimuli or death stimuli. So that is about intrinsic pathway of apoptosis. Thank you.